Hi, this is Professor Tom Webster, and I'd like to thank you for the opportunity and invitation to speak to you on how we are using nanomedicine to treat various cardiovascular diseases. That builds up in your arteries, opening those arteries, maintaining that those arteries are open by decreasing thrombus formation and inflammation around cardiovascular stents, and some of our exciting work developing what we call cardiac patches, or materials that can be placed on top of the part of the heart that dies when you have a heart attack. So, as you can see on this slide, I have several academic appointments where I teach, and also have started a number of companies, some of those related to cardio nanomedicine or the use of nanomedicine in cardiology. So, of course, what we're talking about here are cardiovascular diseases. I don't think I have to spend too much time emphasizing the role that cardiovascular diseases are playing in decreasing human health. In fact, some of you may not know, but the average life expectancy around the world is actually decreasing. And yes, some of this is due to COVID, but a large part of this is also due to traditional diseases like cardiovascular diseases. So I would beg each of you who are doing research in this area, we have got to think out of the box. Our traditional mode of treating cardiovascular diseases is not working. And here are some statistics from the United States where I spend most of my time really highlighting the problem with cardiovascular diseases that seems to be a persistent problem in our healthcare. 1 in every 3 American deaths are caused by cardiovascular diseases. The approximately 84 million people in the United States suffer from cardiovascular diseases, costing our healthcare system here in the US about 300 billion per year, 753 billion worldwide. And of course, these diseases are not going away. We do not have solutions, and the cost of treatment is just going up. The number of people getting infected or afflicted, I should say, is going up too, where many thought groups have predicted in the US dollars 20 trillion to fight globally cardiovascular diseases by the time we get to 2030. So again, we have serious problems with cardiovascular diseases, and our conventional approaches are not working. Of course, one area that I will talk about in this presentation that we have been focused on is myocardial infarction, or a heart attack. So this is obviously one of the most common types of cardiovascular diseases, where you have cardiac ischemia, which is one of the main arteries or branches to from the heart, or an artery becomes blocked. And obviously this causes the heart to beat more intensively to get through that blockage, which can lead to a heart attack. A heart attack does cause cell damage. And unfortunately, this is something we don't have solutions for. Yes, we could put you on blood thinners. Yes, we could do other kinds of treatments. But the fact that cells die during a heart attack is something we have to acknowledge and something we have to find solutions to. So there is cell death that is irreversible for cardiac ischemia. There's regional contractile dysfunction, and muscle tissue gets replaced by scar tissue. Scar tissue is a way that our body tries to temporarily heal tissues, but in some areas you get chronic inflammation where macrophages and fibroblasts will actually produce a type of tissue that is meant to be a temporary tissue, but in the heart, many times that temporary tissue does not get remodeled into cardiac muscle tissue. So clearly it doesn't have the same mechanical properties. And whenever these irreversible problems occur after a heart attack, can lead to another heart attack because the cells have died, cardiomyocytes have died, and that tissue does not have the same mechanical properties as it once did. So cardial infarction is literally the death of heart muscle. So, of course, there have been solutions people have been researching to treat heart tissue, to regenerate healthy tissue where that dead tissue is in terms of post-myocardial infarction. And some of those have involved engineering electroconductive and perhaps antimicrobial cardio patches. So this is where you're using tissue engineering approaches with or without cells to serve as a patch. There have been people who have proposed that you have very highly invasive surgery to place this cardiac patch on the surface of the heart. Our research has been focused on injectable materials that can be injected to and around the damaged area of heart tissue after a heart attack. The material then attaches to that dead tissue and regenerates healthy tissue. And obviously, I use the word electroconductive up here because you really want a material that is not only strong, clearly not only biocompatible and cytocompatible, but also is electrically active. The heart is electrically active, so using conductive materials, strong materials, and biocompatible, cytocompatible materials that can regenerate each layer of the heart tissue, the epicardium, myocardium, and endocardium, is critically important. So we need to think about composite materials. We have to think about bioactive materials if we want to develop this cardiac patch that can be used in the future. There are many problems with approaches that the field has been taking. Primarily, a lot of people have been investigating one uniform type of material, but yet we know the heart is a composite material. It has nerves. It clearly has cardiomyocytes. It has cardiac smooth muscle cells. It has different kinds of cells, different kinds of tissue, different arrangement of materials or proteins like collagen. So a lot of times this composite nature of the heart tissue, of the heart wall tissue itself, is not emulated in what people are trying to propose. Clearly, this can lead to a lack of effective vascularization to occur in that area of the heart. It can lead to the inability to mimic complex architecture and characteristics of that native tissue. And even if it does regenerate a beating part of the tissue, one problem with these cardio patches is this part of the heart that's been newly regenerated beats at a different frequency, beats at a different rhythm than the rest of the heart, which is clearly problematic. So not only have people turned to synthetic materials, obviously to regenerate the heart, but heart transplants have been a way of trying to deal with this problem. There's been a lot of wonderful advances in using animal hearts, especially recently, as well as heart donors to heal the heart tissue. But as an engineer myself, we can continue to wait for us to get enough animal hearts or enough human hearts to be able to transplant and allow that technology to go forward. Or I think a better strategy is really to think of bioengineered scaffolds, cell-based therapies with bioengineering scaffolds as this composite material that can really help damaged heart tissue. I would also say, as an engineer, if we can develop a material to regenerate damaged part of the heart tissue, then we understand from a biological point of view, from a mechanistic point of view, from a cell point of view, what are the best strategies. So this empowers us to understand these properties of a heart tissue, to be able to recreate it synthetically, which is something you don't get if you look towards healthy tissue as well. If you think about, again, the bioengineered cardiac solution, really for a cardiac patch, really most people are now incorporating cells because cells are the best type of material to regenerate a damaged part of the heart tissue. But there are many challenges with cell therapy. There's ethical challenges. Where do those cells come from? And is that source ethically okay with you and the patient. There are viability and phenotype stability problems, ineffective homing of the cells. In other words, many cell therapies, when you inject cells into damaged tissue, they don't stay where the damage is. They will go to where the healthy part of the heart is. But the healthy part of the heart doesn't need regeneration. So you have really an ineffective homing or inability of a cell to stay where the damage is, low cardiac engraftment efficiency, immune system reaction. Clearly, if these aren't your own cells, you run the ability or the problem of the body trying to reject those cells as well as the new material that's been implanted. These can be time-consuming, regulatory, and exorbitant costs. So this is what leads us to nanotechnology, led me to nanotechnology. As I'll show you, we have a history of using nanomaterials as very effective implants in the orthopedic space that has now opened our eyes to using these materials as more effective cardiac patches and for cardiovascular applications. We have been interested in nanomaterials for over 25, 30 years. These are materials on the dimension of 100 nanometers or smaller. They can be particles, nanoparticles, nanotubes, or a surface that simply has a nano texture that resembles the natural texture of the heart tissue itself. Heart tissue is composed of proteins. Proteins are nanodimensional, so therefore it stands to reason and has been proven that the heart itself has a nano texture. So when using nanomaterials, nanofiber scaffolds, we are incorporating nanotechnology with biology and biomaterials for heart repair, specifically in this case as a cardiac patch. We have to be concerned about promoting the adhesion, the migration. When we use these materials alone, the adhesion, the migration, and the function of cardiac myocytes, cardiac fibroblasts. We have done a lot of work, which I'll show you is in vitro, the differentiation of those cells, and then also looking at paracrine factors that are released from these cells to stimulate other cells to regenerate natural heart tissue. As I mentioned, the heart is a composite tissue, so you not only have to be concerned about regenerating cardiac tissue, but angiogenesis, the blood vessels that are found inside the heart, as well as nerves that are found inside the heart. And in some cases, bacteria or infection can be problematic. Anytime you're implanting a foreign material into the body, infection can be a problem. We have seen nanomaterials can reduce infection without using antibiotics. So some of the materials we've been looking at in the study that I'll present to you, gelatin, denatured collagen, very easy, very biocompatible to get. It's a gel. So this enables whatever we put into that gel to be injectable. And again, if we create it correctly, it can attach to damaged parts of the heart tissue, attach to it, and then begin the stimulation of these cells to regenerate damaged heart. Graphene has been our nanoparticle of choice. Carbon-based nanoparticles are very biocompatible. Graphene is conductive, easy, inexpensive to add to these gel-like formulations. And if in a composite form, you can distribute these nanoparticles, or in our case, some of the times we're using nanorods or carbon-based nanotubes, which can be distributed anisotropically to give heart tissue this anisotropic electrical and mechanical property. Remember, the heart tissue is anisotropic. It beats in a certain direction, and it's stronger in one direction than the other direction. So we can use nanofibrous materials like graphene, like carbon, in order to emulate those properties for the more effective regeneration of heart tissue. So electrical conductivity, as I'm emphasizing here, this mimics cardiac tissue. So in my mind, you need a cardiac patch or a cardiovascular material that is electrically active. And I think nanofiber structures are the way to go. So nanofiber structures can emulate that anisotropic electrical property. It can also create a structure similar to the heart extracellular matrix, which again is a way to trick those cells into thinking we're implanting heart tissue itself to regenerate appropriate tissue. Conductivity for electrical propagation of the native myocardium. I already mentioned that there's a preferred electrical conduction through the heart that can only be mimicked through a fibrous or a directional type material. So again, here are some of the materials we're using as the matrix, and here's some of the nanomaterials. CNT stands for carbon nanotubes that we're using and have used in these approaches. And I would, be happy to highlight to you that what we're seeing is very versatile. As long as our material is conductive, graphene or carbon nanotubes, or even we have some research with gold nanowires, we can add it to a number of different matrices, gelatin, synthetic biomaterials. When we create linear patterns of these materials, we can induce anisotropic electrical properties, anisotropic mechanical properties, and get healthy regeneration of the heart tissue. That's what I'll show you. So here's where our history comes from that gave us a lot of confidence that these approaches could be used for the heart tissue. And it's when we have taken the same approach for other implants, mostly orthopedic, but in some cases vascular that I'll talk about, and antibacterial applications. So here's our approach that we have taken is we take medical devices off the shelf. Here's a 3D printed spinal cage. Here's an amputee, in this case, as a cardiac patch. We have to be concerned about promoting the adhesion, the migration, when we use these materials alone, the adhesion, the migration, and the function of cardiac myocytes, cardiac fibroblasts. We have done a lot of work, which I'll show you is in vitro, the differentiation of those cells, and then also looking at paracrine factors that are released from these cells to stimulate other cells to regenerate natural heart tissue. As I mentioned, the heart is a composite tissue, so you not only have to be concerned about regenerating cardiac tissue, but angiogenesis, the blood vessels that are found inside the heart, as well as nerves that are found inside the heart. And in some cases, bacteria or infection can be problematic. Anytime you're implanting a foreign material into the body, infection can be a problem. We have, in contrast, over here, the spine, and then one is in the foot. So this case are pedicle screws. These are simply screws that are used in the spine. We have an anodization process that creates nanotextures on these titanium-based materials. We've been implanting these since about 2016, 2017. In the United States, if you have a failed implant, if you ever want to check if somebody's implant has failed, a manufacturer's implant has failed, you can go to what's called the MAUDE database, MAUDE, and then type in the name of the company, the name of the type of implant, and you can see how many have failed. In this case, none have failed. In the 6 or 7 years, I suppose, that we've been implanting these in about 14,000 patients so far, we've had no cases of infection, no cases of loosening of these implants in the spine, no cases of extensive inflammation, chronic inflammation, just success, complete success of these materials, which we attribute to the nanotexture that we created. Without a nanotexture, the conventional pedicle screw will fail about 5 to 10% of the time. It loosens or gets infected or wears. There's a number of failure modes that can occur for these devices, but it usually occurs 5 to 10% of the time. And even in cancer patients, bone cancer patients who get these implants, you can have a failure rate up to 60%. So the fact that we were able to eliminate failure due to infection, failure due to inflammation, any kind of failure, simply by putting a nanotexture on these implants, which, by the way, here's what that nanotexture looks like right here. The anodized surface is, quite successful, I believe. Another device is silicon nitride. These are spinal fusion devices. Also used in the spine, we have a laser etching process that created nanotextures on those types of materials, and over 4,000 have been implanted with no cases of infection as well. The last one that I can mention to you that were in humans is foot screws. These are metal-based foot screws coated with calcium phosphate. So we coat them with nanoparticles of calcium phosphate. They provide a nanotexture, and we've seen 0 cases of infection. Over 1,500 have been implanted. Importantly, these nanotextured foot screws are made for diabetics. So they're made specifically for a portion of the population that has a higher chance of getting an infection. Diabetics have a higher chance of getting an infection than the rest of the population. So we're really testing the ability of these nanotextures to promote bone growth, to reduce infection. And again, I could spend a great deal of time. Why are we seeing these things? The field, we're not clearly the only people who are looking at nanotechnology or nanotexturing of implants, but the field has really highlighted some fundamental scientific reasons why these surfaces work. The first very generic reason is nanotextures are everywhere. They're on cicada wings, locust wings, lotus leaves, gecko feet. And the nanotextures on those natural materials or living organisms have a special function, right? A lotus leaf enables a water drop to go into the center of the leaf so that the plant accumulates water for life. In this case, the insect has nanotextures. Here's an electron microscopy picture of those nanotextures on the wing of the insect, and it repels bacteria. So a research group recently mimicked the nanotexturing of this insect wing, as you can see here. They put bacteria on the surface, and they found that bacteria do not grow on this surface. In a more scientific mode, researchers from Australia have shown biophysical models where bacteria membranes are too stiff to fit into all the contours of a nanotextured device. In contrast, cells like cardiomyocytes or osteoblasts, bone-forming cells, they're much bigger, 50 microns compared to 1 micron here, and the mammalian cell membrane is much more flexible, and it prefers a 3-dimensional architecture like seen here because it resembles more of a natural tissue. So it attaches more, grows more, and produces tissue more on those nanotextures. But that does not happen with a bacteria, again, because the bacteria membrane is too stiff. Surface energy is much different between these two materials. Here's a nanotextured polymer surface and a micron textured polymer surface. And just to highlight, here's a nanotextured type surface that was created to be hydrophobic. You can see the water drops beating up. You can simply change texture, change the height, the width, the length of that texture, and instead of being hydrophobic, it's now hydrophilic. And you can see the water drop here being soaked in to that surface. This is all a result of nanotextures, not changing chemistry, just nanotextures. Well, obviously, if you change the surface energy of a material, you change what proteins absorb. Some proteins are better for cardiomyocytes to attach than others. Some proteins are better for bacteria to attach than others. Some proteins, like IgG, if that absorbs to your surface, it will promote macrophage function and lead to inflammation. So this study first is just showing one particular protein, fibronectin. Fibronectin, if it absorbs to your surface, is a great protein for cardiomyocyte function. And you can see, first of all, by creating a nanotexture, you get a lot more fibronectin that has absorbed than on a micron textured surface. Secondly, the geometry of fibronectin is different on a nanotextured surface than a micron textured surface. In fact, the texture creates a more fibrillar, nanofibrillar fibronectin structure. This is more bioactive. It exposes more amino acids for cardiomyocytes to attach to. In contrast, over here, the fibronectin absorbs like a hemispherical orientation, which is not bioactive. So you can use nanotextures to absorb the proteins you want to get the cell response you want. And incidentally, it can also make the protein more bioactive after it's absorbed. We've created an equation that helps you model this. And basically this equation, if you give us an implant, like many people have, we can determine what size of nanotexture to put on your implant to, in turn, create a more bioactive surface. Okay, so let's cover here some cardiovascular type stuff. Just a quick study on stents. I'm pretty sure we're all familiar in this conference with what stents are. You have a clogged artery One of the very easy, well-accepted approaches today is insert a stent, usually through your femoral artery, go to one of the arteries close to your heart and have this as a shape memory, nitinol alloy, for example, and then you expand the stent to prop open the blood vessel so it allows blood to flow. Unfortunately, the body wants to get rid of this metal. So a critical criteria for this stent to be successful is cells have to cover this metal, healthy cells. And the cells that line the inside of your arteries are called endothelial cells. So if you can design a stent where the inside of the metal structure is covered with endothelial cells, then it masks this stent for the inflammatory cells that are going by, like macrophages, monocytes, platelets, white blood cells, other cells that can attach to the stent. And if they do, they will secrete proteins to recruit more of those inflammatory cells. So the goal has been to get endothelialization, coverage of this stent with endothelial cells. And if you can do that, if you can do that, you have a better performing stent. If you don't do that and your stent remains open, and here's a picture of a stent that we used in an in vitro study with endothelial cells, and you see after 24 hours how few endothelial cells have attached. So all those open areas are prime spots for macrophages, leukocytes, neutrophils to attach to and basically signal through a chemical mediator that a foreign stent has been inserted, which could lead to inflammation or thrombus formation, basically buildup of plaque. Again, that you just tried to get rid of and push to the sides, you now have more plaque coming back because of these open areas. In the same study, we created nanotextured features on titanium. By using a anodization process, we created linear patterns of those nanotextures indicated by these arrows. The size of those features are indicated below each fluorescent image. And you can see that by going from a large feature size down to smaller, we were able to cover more of the stent with endothelial cells. And that creates for a more effective stent, a stent that is less recognized by your inflammatory system to reject it. So this is another example of how nanotexturing can be used in the cardiovascular space. Don't change the chemistry. You don't have to change the implant procedure. All you're doing is taking the same stent that has been commercialized and used for decades and create a nanotexture that can reduce chances of thrombus and inflammation and increase the embedding of that stent into the vessel wall to prop it and keep it open. And keep in mind, all of these things that I am mentioning were done without drug release. One of the big problems with stents about 15, 20 years ago were drug-eluting stents, where companies were putting drugs in stents, but unfortunately, some of those drugs that were released were not released correctly and led to patient death. So we are always looking for non-drug approaches to health, and this is one, nanotexturing, that can simply increase endothelialization, decrease inflammation and thrombus without resorting to drugs. As I mentioned, we work with a lot of companies. So if you're interested, please send me an email. We can work with you. We have a term, a stamp we give you called nano optimized. So after this nano optimization of your medical device is complete, we test it. We then can give you a certification that your implant has been nano optimized. So now moving on to cardio patches, same idea. We can use these nanostructures. Here's graphene nanorods. But as I mentioned before, we've also used gold nanorods, which are also conductive, carbon nanotubes, which are conductive. We used a series of conductive nanomaterials and then combine them with a gel matrix. And here I'm going to show you results on gelatin, but we've also used Gelma. We've used other kinds of hydrogels that are currently being used in medicine. Make a composite out of those materials. You can electrospin these materials into an appropriate patch orientation. This is also during electrospinning when you can align the graphene nanofibers or nanoribbons to basically make that anisotropic electrical, anisotropic mechanical properties. So during electrospinning, you can actually make a larger device that has these attractive anisotropic properties. We, of course, have characterized these that I'll show you results for electrical properties, for mechanical properties to ensure that they meet standards. We have completed results on in vitro and some animal studies showing that they can regenerate healthy tissue in the area of damaged tissue. And then, of course, we've looked at a lot of biological factors. What are these cells producing? Are they recruiting other cells? How are they performing in the heart? So here's some scanning electron micrographs of what these graphene nanoribbons with gelatin look like, and you can see the different weight ratios of the as-spun fibers. We have good control over the diameter of these fibers. We, sometimes look towards matching the diameter of a collagen fiber. Remember, collagen fibers are larger, and then collagen fibrils are smaller, so they have a hierarchical arrangement. But we have pretty good control through this electrospinning process of the size of these fibers, no matter what weight ratio of graphene nanofibers that we add. So here's a graph showing you some of the various fiber diameters we can get at different graphene nano ribbon concentrations. Here's some FTIR spectra confirming the chemistry, and again some measurements confirming the exact diameter that we get. So pretty good control, narrow distribution of a lot of those diameters. We also, because this is an injectable that we want to inject into the heart tissue, we look at swelling ratio, so different weight ratios of graphene nanofibers, swelling time. So we have pretty good control over that swelling ratio, anything from just below 100% up to 300%, depending on what is needed for the size of the heart damage that occurred after a heart attack. We also can look at how the swelling or how degradation occurs. So these materials degrade. Obviously, gelatin degrades. There's also some limited evidence that we have that carbon in the form of graphene nanoribbons can also degrade into the heart. So we have pretty good characterization, and we can tweak this degradation rate to match the growth rate of the cardiomyocytes in the new heart tissue. Again, some scanning electron micrograph pictures. Here's some surface energy. As you remember, surface energy is something that's been very important to my research group to optimize protein interactions, protein concentrations, to give us the best kind of cytocompatible material possible. So we've done a lot of those measurements, and again can confirm what the exact surface energy of our patches are and how that relates to increased protein absorption as we get more and more graphene nanofibers. Mechanical properties are right where they need to be. So looking at tensile strength as an example, elongation at break, tensile modulus, the gelatin graphene nano ribbons have the mechanical properties that are close to that of heart tissue itself. Electrical conductivity is also pretty similar to natural heart itself. Here's some conductivity measurements in various media where the conductivity in Siemens per meter, you can see that we get an increased conductivity. And then interestingly, when we add more past 0.1 weight percent, we start going down. This is because some of those nanoribbons agglomerate into larger structures. So we're losing some of the electrical conductivity, but nonetheless, they're still conductive enough for heart tissue applications. And that conductivity maintains with time of culture in cell culture media. So we're pretty happy with all of those, but most importantly, the cytocompatibility results. These are cardiomyocytes that were cultured with cardiac fibroblasts, a co-culture system. They were seeded on a plain gelatin and then a gelatin with 0.1 weight percent graphene nanoribbons and 0.5 weight percent graphene nanoribbons. You can see that there are concentrations in which we start getting a slight reduction in the cardiomyocyte cardiac fibroblasts viability. So when going from 0.1 to 0.5, you can see in the live dead assay, we are seeing a couple more dead cells, which we don't want. So we've often stuck to our animal studies for this 0.1 weight percent graphene nanofiber in gelatin patch, but you can see they do grow on those samples with time. In fact, day 1, day 4, day 7, the cells are continuing to grow on the surface, continuing to spread out and look very, very healthy. And again, this is on the 0.1, our optimal material formulation that we've made so far, gelatin graphene nanoribbon patch. So let me also highlight to you that we've looked at certain proteins in the interest of time, which I think I'm running out of. Let me just show you a movie. This is our culture, which you can see the volume a little bit, the culture of the gelatin with graphene nano ribbons that we've created. The cardiomyocytes with cardiac fibroblasts are seated, and you can see the beating that is occurring after just 10 days post-seeding. We actually see this beating occur much earlier. And on the control samples, gelatin samples only, which don't have the graphene nano ribbons or any conductive material, we don't see beating that occurs at all. So we're very happy with this whole patch beating phenotype. We've also looked at the electrical activity, and you can see that we're getting relatively good electrical activity. Remember, one of the problems with, for example, pure gelatin is you don't get a normal beating that occurs, and you need a beating that occurs similar to the natural heart itself. So we're able to get that when we have 0.1 graphene nanoribbons with our gelatin. So we've also looked at factors for angiogenesis. As I mentioned before, heart is not just one kind of tissue, it's a composite tissue. And when we do our cell studies, we do see number of vessels that are growing onto our material when we seed endothelial cells, HUVEC cells to be exact, where you can see our 0.1 formulation has a high number of vessels that are growing, a number of new vessels that are growing, and certain proteins like CD11 that we can measure, or I'm sorry, CD31 that we can measure its coverage in terms of how well angiogenesis is occurring. So I hope I gave you a good idea of our cardiovascular nanomaterials that we're looking at: nanotexturing stents, cardiac patches, which can be used to heal damaged heart tissue. But clearly, we're not alone. There are so many other cardiovascular nanomaterials that people are using. This is a review paper that we had written where the polymer that's being used here, everything from PGA to PEG, combined with different proteins, different nanomaterials, they're all showing an improved ability to promote vascular function and heart function. Carbon nanotubes are seeing a lot of attention in this field. We're looking at endothelial cells, fibroblasts, or cardiomyocytes, and a PLGA, carbon nanofiber composite material, is showing high amounts of, with electrical stimulation, some of the highest number of change in cell growth you can see for the highest amount of carbon fibers that were added. So I hope during this presentation I gave you an idea of how nanotexturing can be used in orthopedics, eliminating the risk of implant failure completely, and now moving into cardio nanomedicine, using these for stents, using these for cardiac patches, using these for many applications in which you can use the nanomaterial to give you good mechanical properties, good electrical properties, and ultimately better cytocompatibility properties. I, of course, am open. I want to thank my lab group and my funding agencies, and here's a nice structure we created. This is out of graphene nanoribbons and gelatin. We 3D printed it into a thumb, and you can hopefully, through this image, see it beating. So this is, what indicates to us a good cardiac organoid at day 27 is we have good knowledge of these systems in order to create in vitro a beating heart. So thank you. I appreciate it. Please email me questions if you want to collaborate, Thomas_Webster@brown.edu. And thank you again for the invitation.